Good day. Today we are going to investigate the fine art of identifying post-translational modifications from MSMS data via bioinformatics. So I realized it had been a few years since I filmed a video on this, and a lot of stuff has actually been going on in the meanwhile. So today we're going to talk about some of the different ways that we use algorithms to reveal post-translational modifications. We're going to start with kind of an oldie but a goodie, the approach that allowed us to adapt standard database search engines to look for variants of amino acids, not just the amino acid masses themselves. From there, we will talk about kind of a villain's gallery to look at all the different mass shifts that seem to crop up in lots and lots of data sets. If your favorite modification is missing, feel free to leave a comment about that. And don't forget the slides for this talk are available from a link in the description below this on YouTube. Okay, so from there we're going to talk about a rather new approach called open search that's become really in vogue for finding sets of modifications. We'll try to talk about some of the limitations of this algorithm that may hold back some of the promise that it offers. But I hope that you'll come away as a more seasoned user of the open search approach. And from there, we're going to move on to two big problems that always seem to crop up when we talk about post-translational modifications. The first of those is, how do we control the false discovery rate for modified residues, or for modified peptides? And then we will talk about, how do we localize them? How do we make sure that this mass shift appears on this particular amino acid? So I think many people have seen this diagram now. If you've already watched the lecture on proteomic identification, this should come as no shock. But I want to spend a little time talking about this post-translational modification expander. There are two original papers for seaQuest. The 1994 one sometimes gets cited, and we often see people citing the 1995 one. The difference between these two papers was the ability to recognize post-translational modifications from our data. So the 1994 seaQuest was fine for identifying peptide sequences, but the 1995 seaQuest was necessary if you were going to go looking for things like methylations or oxidations or whatever. So this PTM Expander really bears a little bit of consideration, the process of starting with a set of peptide sequences to compare and changing to a set of decorated peptides, ones that have post-translational modifications assigned to particular positions. I believe people have seen this slide if they saw the protein identification one, but I want to walk through it a little bit. Imagine that we have this peptide in the sequence database. In this case, it's from alpha-casein in cow's milk. So we see that DIGSESTEK looks like a perfectly ordinary peptide. Typically, when we go searching for phosphorylations, and certainly there are lots of phosphorylations in cow's milk, you could specify serine maybe at its normal mass of 87 Daltons, or it might be 80 mass units higher to reflect that it's a phosphoserine. Similarly, threonine could be 101 mass units, or it could be 181 mass units if it's bearing a phosphorylation, a phosphothreonine. There's also the case that tyrosine might be present, and that tyrosine might be in its ordinary mass, or it might be at 80 mass units higher. So any time the software encounters a serine, a threonine, or a tyrosine, it has to consider a fork in the path. That this peptide DIGS up to the S is just one path, because none of those three letters, D, I, or G, are modifiable. But S is. So when it hits this S, it must fork the path. When it hits this E, it just extends each of those paths further. When it hits this S, each of those paths in turn must split. So now we have 4 paths that we must follow. T gives us another fork on top of the 2 forks already there. Now we have 8 paths that we must follow. E, K, no divergence further. But we see that this expansion of search space is going to fork every time it hits an amino acid that is modifiable to 2 different paths. Now I would note that some people who do really advanced kinds of phosphosearching consider actually 3 possibilities for serine, threonine, and tyrosine. It could be that we have something called beta elimination that causes the phosphoserine to throw off not a phosphorylation, but phosphoric acid. So it may have gained 80 to become phosphoserine, but it loses phosphoric acid, losing 98. So in effect, instead of the normal mass of 87 for serine, we had 167 as we gained phosphorylation, but when we lost phosphoric acid, we dropped below 87, the mass of serine, to create something called dehydroalanine. So a loss of water from serine creates dehydroalanine, a loss of 18 mass units. That's the same thing as if the serine became phosphorylated and then threw off a phosphoric acid. So so you could consider, in fact, 3 possibilities for each serine. We have serine at its ordinary mass, serine for its phosphomass, or serine for its dehydrated mass. The same is true for threonine, but thankfully not for tyrosine. So if you were to consider that, you would actually have 3 different paths emerging from every serine and every threonine. That's an even harder kind of search to compute. So how much more expensive is it to go from an ordinary serine search to a phosphoserine search to a phospho or dehydro serine search? The way that we work this is to consider how many possibilities there are for each modifiable residue, in this case 2, phospho or not phospho, how many positions within the peptide are modifiable in this way, in this case 3, because there's 1, 2, 3 amino acids that are modifiable here. So 2 raised to the 3rd power is 8, which is to say this one peptide becomes 8 peptides in that search. How much worse does this get if we consider all 3 possibilities for each serine and each threonine? Well, we don't have any tyrosines to worry about in this peptide. So that would mean that we have 3 different masses that we must consider for each of those amino acids at 3 different positions. 3 to the 3rd is 27. So this one peptide becomes 27 peptides. So the first thing that most people think is, gosh, that's why PTM searching takes so long. And you're right, that's why phospho searching takes so long. But the other thing I would consider is that it also means that the algorithm must now discriminate away more wrong answers for each spectrum to get to the one correct answer that exists in the sequence database. So our discrimination also gets harder. The other thing I'd have you mention, and this is kind of an odd point, is that serines and threonines are not spread uniformly throughout the sequence database. Instead, we find that sometimes we see runs of serines and threonines in proteins, and in the rest of the protein you have relatively few serines and threonines. In that case, what you're going to see is that peptides that contain serines and threonines have an advantage over peptides that don't contain serine and threonine, because they get compared in a lot more forms to spectra than they would in a non-phosphorylation search. So we have more time, we have a greater challenge to discrimination, and we have a bias, in effect, for peptides that contain modifiable residues. So what kinds of modifications do we search for? Are PTMs all about phosphorylation? Well, if you read the literature, you might think so, but in fact we see that there's a lot of different modifications out there that we consider. In fact, there are hundreds. If you go to a site like UniMod.org or Resid, or kind of the old Delta Mass resource that was assembled by ABRF, you will find that there are huge numbers of potential modifications for us to consider. The UniMod resource is really helpful in that you can simply write in the modification you're looking for, click Show All, and up pops a list. You'll frequently find that you get many hits on one of these. If you, for example, knew that somebody included a plus 57 in their sequest search and you wanted to understand what that was about, you could just type 57 in here and say Show All. But I tell you, anything that includes the digit 5 followed by the digit 7 is going to show up in that search. So the search engine's a little hinky on that score. So here we see that UniMod has been around for a long time, but has a very well-attested set of modifications for people to think about. Once you click into these, you'll even see that there are publications strapped to each of these to say, this is the publication that helped us to establish what chemistry is going on here, how many oxygens or carbons or nitrogens or whatever are involved, and so on. So it's a really great resource for that. Those papers are, by the way, worth reading. So what do we get in our rogues gallery? So I would start by noting that a lot of the modifications we search for in proteomics are not interesting. And I don't mean to say this in a way that's dismissive. I mean it to say that what we're trying to do is account for the fact that the chemistry we use, the biochemistry of manipulation of these samples to get them into the mass spec, often leads to a change in their chemistry. So we should start with the case of cysteine. I think a lot of people realize that when we do proteomics, we very often break apart disulfide bridges between pairs of cysteine residues, and then we alkylate those free cysteines to prevent them from reforming their bridges. So one of the most common ways that we alkylate them is iodoacetamide. Iodoacetamide creates a modification called carbamidomethylation. It's kind of a mouthful, and it's not to be confused with carboxyamidomethylation. That produces a shift, a monoisotopic shift is what I'm showing here, of 57.021464 Daltons, which means that every cysteine, instead of having a mass of 103, has a mass of 160. All right, that is not, however, the only way that people mess with their cysteines. We frequently find that people who are using iTrack reagent to make a quantitative proteomics experiment will, instead of using iodoacetamide, use something called MMTS, and I'm not qualified to pronounce what that stands for. All right, that creates a modification called beta-methylthioylation, which has a mass shift of very close to 46 Daltons. So instead of having a mass of 103, it's going to have a mass of, do my math very quickly in my head, 149. Okay. So cysteine gets handled a variety of different ways. There are another two reagents that came up in our Twitter discussion of how to make this slide. Iodoacetic acid is probably the most common one after these two, but there are lots of ways that people mess with cysteines. And cysteine, I got to tell you, cysteine is shifty. If you're just looking at it out of the corner of your eye, you might notice it's changing its chemistry and getting formulated or something bizarre like that. So cysteine's tricky. All right, next up, methionine. Cysteine, methionine, tryptophan, these are the three least common amino acids, and yet we see that methionine is tricky in its own right because if in handling your sample, you might leave it on the bench a little while and you find that methionine has become oxidized. So oxidized methionine changes its mass by 16 mass units. And if you remember your table of atomic masses, you're going to remember that on the periodic table, oxygen shows up at 16 Daltons. Okay, great. So next up, N-terminal glutamine. Not a lot of people may remember this one, but when you have a Q at the left end, the N-terminus, the left end of your peptide sequence, the side chain of glutamine can swing around and attack its own N-terminus, and it loses mass rather than gains it. Now again, this isn't telling you a whole lot about the biology of your sample, but it's important to keep these modifications in consideration because depending on how you've treated the sample, these may be the dominant sets of PTMs that we recognize in our sample. So these are chemical modifications, not necessarily informative about the biology of the sample. Okay, we mentioned isobaric labeling for quantitative experiments. If you must consider these, remember that the iTrack and TMT reagents do not just alter the N-terminus of the peptides, they attack primary amines, and primary amines are also found in, a primary amine is also found in a lysine side chain. So if you do an iTrack or TMT search, you'd better remember to put it not just on N-termini, but also put it on lysine. And of course those two have different masses, plus there are other isobaric tags out there. iTrack is also available in an 8-plex form, for example. Okay, there are some other things that can go wrong as well, though. You may be using urea that a grad student prepped 3 months ago or 6 months ago, and heaven help you if so, because there might be a degradation product in there that can attack your lysines to produce carbamylation, so your lysines gain 43 like that. So every time we modify a lysine, we should remember that we may not just be adding mass to the lysines, we may also be blocking that lysine from participating in a peptide cleavage event. So your protease, trypsin, may not chop after a lysine if it's been modified in particular ways. That's a big deal. Okay, the last one someone reminded me to mention is deamidation. So it might be that you have a motif of asparagine followed by glycine in your peptide. There's a weird succinimide mechanism by which the side chain of the asparagine can attack the glycine, and lo and behold, you end up replacing the asparagine, in half the reactions if I remember, you replace the asparagine with an aspartate, which means that you gain one mass unit by just the chemistry sort of blinking on you. Now I note that these are kind of canonical mass shifts that we all think about, but sometimes they show up in other guises. So I would first start with iodoacetamide because it is so widely used. There are some people who put rather a lot of iodoacetamide in their samples, and they may leave it to marinate in that iodoacetamide a little long, and this can lead to an carbamidomethylation. And in a case like that, instead of just getting acetylation of the side chains of cysteine, you also can get a modification on the N-terminus of the peptide. Obviously, that's problematic. Okay, similarly, we think of this deamidation as just a spontaneous process, but a lot of people who are studying glycans, peptidoglycans, will use something called PNGase F as an enzyme to lop off a complex carbohydrate, but it lops it off in such a way that it leaves a marker of its location. That asparagine becomes an aspartate. So it might be that somebody who's doing a deglycosylation experiment is looking for where the enzyme did its work by where it gained this mass unit. Of course, most people who are doing post-translational modification searches are not just trying to deduce something about their chemistry, they're trying to learn about the biology of the sample and the post-translational modifications that it contained. And here you might be tempted to say, oh, here he's going to rattle on and on about phosphorylation. I already did that, so I'm not going to repeat that here. I would note, however, that acetylation is a very big deal. This acetylation can be taking place on N-termini, of course, but it may also be found, especially on protein N-termini, after lopping off the initial methionine, but it may also be found on lysine side chains. Certainly when we do stuff like examine the histone code, the way that lysine and arginine can be modified in that context is extreme. So being able to look for acetylation at 42 mass units is quite important. Not going to talk again about phosphorylation, but hydroxyproline is definitely worth talking about. If you are dealing with solid tissues, like biopsies, it's quite likely that you have a large amount of collagen in your sample. And collagen is absolutely littered with hydroxyprolines. If you do not include that in your database search, you're not going to see it. So you better include hydroxyproline if you're doing solid samples of that sort. Now, some friends on Twitter reminded me that it's not just solid samples that can contain collagen. Urea and cerebrospinal fluid have also been shown to contain a fairly high amount of collagen. Any sample that contains large amounts of extracellular matrix is going to show you plenty of hydroxyproline. So that's a proline that instead of having a mass of 97 has a mass that's 16 higher, which comes to 113. I want to point out something. It might seem to you that if you see a proline, that a proline has gained 16 mass units, if you don't have your chemistry thinking hat on, you might say to yourself, oh, in this case the proline has mutated to become a leucine, because leucine also has a mass of 113, or isoleucine also has a mass of 113. So it's important to remember that in lots of cases, inferring what these mass shifts mean is going to require that we think about different possible mechanisms that may lead to that mass gain. Okay, methylation is a very, very big deal. One of the most obvious places to find methylation is on trypsin, because by methylating and dimethylating trypsin, we produce an enzyme that's more specific. So in a case like that, you're much more likely to find 14s or 28s that represent single or double methylations. But I would note that if you're dealing with formalin-fixed paraffin-embedded samples, it's very, very important that you look for methylation, because one of the key effects of the pickling process is to get lots and lots of methyl lysine. So be sure to look for that. I was not actually aware, but it's quite common for especially neuropeptides to be amidated, to have an amide on the C-terminus of the protein. Sometimes this happens by having a glycine added there and then chopped back a couple atoms to give an amide on the end, on the C terminus, which seems completely in reverse of my thinking. So this is caused by an enzyme called PAM. I'm not going to go into it right now, but its product is to add a glycine and chop it off to produce a mass shift of negative 58. Keep in mind that the sign is really going to matter in these two cases. Also, you might see that people have incorporated a mass shift of 114, especially if they're doing ubiquitin studies or simulation studies. I always need to pronounce that correctly. In both of these cases, a peptide is stuck onto lysine residues as kind of a way for the cell to mark this protein for death. So in a case like that, when we use trypsin, it cuts after a lysine in that added molecule, leaving gly on the primary amine of the lysine, which leads to a mass gain of 114. So these are just a very few of the stories that we would find in UniMod, but I think it's really valuable for everyone in mass spectrometry to be able to recognize what a mass shift of 42 means, not just a mass shift of 80. It's also commonly the case that students don't realize that different amino acids have very different probabilities in a given protein sequence. If you were under the assumption that every amino acid is just as likely to appear as every other, then you would expect sequences to be 5% leucine and 5% cysteine and 5% proline and so on. Biology doesn't work that way. Instead, what we see is that some amino acids are much, much more frequent than others. This graph was taken from the UniProt release notes, and we see that this big bar represents leucine. Leucine by itself is about 10% of all amino acids in proteins. Leucine is a big hydrophobic, so you might imagine it shows up quite frequently in regions that are burrowing through membranes, for example, because that's a hydrophobic region. So leucine is about twice as common as an ordinary amino acid would be in our background model. I already mentioned that cysteine, methionine, and tryptophan are some of the least common amino acids as well. Well, this has an impact on database search for post-translational modifications. Because if you have an amino acid that shows up all the time and you allow it to be modified, you have a much greater chance of grievously impacting your search time and making it much harder for discrimination than if you modify a rare amino acid. So let's consider one of the most common amino acids out there for PTM searching, oxidized methionine. Methionine is one of the rarest of the amino acids. So considering it being present in oxidized and unoxidized form has a relatively low cost. We would be in a much worse place if leucine were found in oxidized and non-oxidized form, because there are so many leucines in a given peptide that the search space would blow up. Okay, so when we think about common amino acids, thinking about leucine, alanine, glycine, valine, that's a very likely place to go. The rare amino acids are C, W, and M. Frequently, we find that the major reason people don't do post-translational modification searching is that it takes a long time, and they think to themselves, I don't want to have to deal with how much time is added by that. So I think it's worth quantifying that. I'm going to start with an assumption here, and I want you to go along with me on it, which is to say, if I have to compare a spectrum to more sequences, I'm going to have to invest more time. And it's going to scale linearly. So if I compare 1,000 sequences to each spectrum, that's going to be 10 times faster than if I compare 10,000 sequences to each spectrum. Now, we're going to talk about a big way that gets violated when we move on to open search. But for now, when we consider just standard narrow searching, I want you to keep in mind that the more sequences you compare to each spectrum, the more time is required. So I've got two different models that I'm comparing here. In both cases, this teal represents no modifications and fully tryptic peptides only. So this is the amount of time, or the number of candidates that must be considered in that very similar case. And these two blue dots are the same size. On the left, I'm considering 3 scenarios that show up for people who have lots of computing power to throw at the problem. I, for example, am a big advocate for semi-tryptic searching, which is to say the peptides need to, are allowed to start at something other than the residue after lysine or arginine, or the peptide is allowed to end at something other than arginine or lysine. So if we do semi-tryptic searching, we get to this magenta size. You can see that that's already something like a 10-fold increase in the number of comparisons we need to make to each spectrum than before in fully tryptic searching. It may be that somebody is searching for a set of peptides that don't correspond to trypsin cut sites at all. If you're looking at native peptides in samples, it's quite unlikely that those are coming from a trypsin-like enzyme. So this unconstrained search is looking for any peptide that starts at any position in the sequence database and extends to any position in the sequence database. For that one sequence. And you can see that the search time required for that is quite a lot larger. That big green circle compared to this tiny teal circle is quite a big boost. Alright, the same can be seen when we look for post-translational modifications. I'm considering only 3 comparisons here though. We start with no modification searching, that's our teal circle. We've got our magenta circle representing the case where we look only for the oxidation of methionine. This is a very common way to do searches. So we see that the cost of looking for methionine in 2 different masses is not all that different. However, once we allow for serine, threonine, and tyrosine to gain 80 mass units on top of methionine being able to gain 16 mass units, we see that our search size has increased quite a lot. So anytime you create a new configuration to search for post-translational modifications, you always want to think about how big am I making my circle? If I start considering 4 different mass shifts all within the set of, in the scope of one database search, the circle that you produce is going to be massive and it's going to have a huge consequence in search time and in discrimination. And it's also going to have this problem of biasing towards peptides that contain modifiable residues. Alright, with that, I'm going to suggest that you take a brief moment to drink a cup of tea and come back for the second part of this talk. And welcome back. From here, I'd like to talk a little bit about more advanced methods for hunting modifications. Now, having invested about 10 years of my life in getting sequence tagging working for recognizing modifications, I feel I would be remiss to leave it completely out of this talk. So this is my one slide on that topic. I hope you'll let me, allow me to talk a little bit about it. Sequence tagging was a method invented by the Matthias Mann lab back in 1994. I spent a good bit of time, as did people at University of California, San Diego, and elsewhere, at getting sequence tagging working really well for peptide identification and for modification identification. So how is it that sequence tagging differs from database search? Sequence tagging is going to rely upon the ability to infer part of the sequence directly from the spectrum itself. So rather than relying on the database as a source of all sequences to be compared to the spectrum, sequence tagging attempts to infer a part of the sequence directly from the peaks observed in the spectrum. So in this beautiful ion trap spectrum, we can see that some of these pairs of peaks are separated by the masses of amino acids. 798 minus 685 is 113. 113 could be interpreted as isoleucine or leucine. This mass shift down here is 57 mass units apart. That could be interpreted as glycine. We might have a pair of peaks to this little stubble down here that represents a mass gap of 87 mass units. That could be serine. So we see that we could walk from peak to peak across this spectrum and infer a partial sequence to give us some purchase on identifying what the rest of the sequence means. So figuring out which gaps are best to chain together to create these tags is the problem of sequence tag inference. The second part of this is interpreting that sequence tag in the context of the sequence database. We have these peptides that we draw from the sequence database. Some part of them may match 3 letters that we've seen from this 3 amino acids that we've inferred from this spectrum. And it might be that one side of the peptide sequence drawn from the database matches to the amount of mass missing on that side. But how do we interpret a mass mismatch on the other side of that tag? That might be the result of a post-translational modification. It might be the result of a change in the sequence. Or it might represent something else. It could represent a false positive, that this spectrum has been falsely interpreted, that this one sequence tag is not real sequence. It's just a set of peaks that coincidentally happen to be amino acid masses apart from each other. So I frequently called this process of interpreting a sequence tag and the spectrum against the observed peptide in the sequence database as the process of tag reconciliation. Sequence tag inference is followed by sequence tag reconciliation, and that might give us the complete sequence of the peptide along with the possibility of a modification. In the end, a lot of people found that sequence tagging was just different enough that it was really difficult for them to understand how the results from sequence tagging could be turned into better database searches. Most people feel very comfortable in the database search paradigm, and thus sequence tagging largely fizzled out. There are a lot of papers published in the space, and the labs that have developed a lot of papers in that space still find it a very useful technique, but I think a lot of people have found it so different that they don't know how to interpret it. So, OpenSearch represented an effort by Chick et al. From the Steve Giggy lab on how to modify database search to make it more amenable to the recognition of modifications that aren't specified in advance. You remember when we looked at database search as a PTM finder, we noted in advance which amino acids were going to be allowed to gain mass or not. And so we had to pre-specify all types of modifications that search would consider. With OpenSearch, we no longer have to do that. Now we're allowing any mass shift, really, to be observed between a database peptide and the precursor that this tandem mass spectrum represents. So if we return to our original figure, we see that the peptide mass filter, which was so very important for narrow database searching, becomes rather less important in the world of OpenSearch. Before, we might have allowed the precursor ion, the peptide ion that we shattered to produce this spectrum, to differ from the peptide sequence by 20 ppm, for example, which is a very, very small amount. It could be 0.002 Daltons, very small narrow window. But with OpenSearch, we typically allow this precursor mass filter to be very, very wide. In the Chick et al. Paper, they considered possibilities of up to a 1,000 Dalton mass difference allowable between the precursor ion that was blown up and the peptide mass that was computed from the sequence database. This is a pretty extreme case. Today, you'll more likely find people using plus or minus 100 or 200 Daltons as a much narrower focus to which ions may be allowed to match. But what we get then is basically where before a tiny fraction of all decorated peptides were compared to a given spectrum. In this case, pretty much all decorated peptides get compared to all tandem mass spectra at a window of size of 1,000 Daltons. So this is a really, really different paradigm. Before, maybe we were comparing 1,000 peptide sequences to a spectrum, now maybe millions. So it's it is quite a substantial difference from it. So how can we get away with that? I would note that at least until about 5 to 10 years ago, people frequently had an accurate mass for the peptide ion, but they had inaccurate fragment measurements. Something like an ion trap, meaning that we saw an ion here in the tandem mass spectrum and we knew its position to within 0.5 m/z, which is not all that accurate, right? But today, a lot of the data sets we work with either come from QTOF instruments or they come from Orbitrap style instruments like the Fusion and so on, using HCD. These tandem mass spectra are very, very high resolution and give us a very certain measurement of what this m/z is for this fragment ion. That means that our ability to use fragment ions as high discrimination engines in their own right, to say that we know these fragments and no others are observed for this tandem mass spectrum, and we know their masses very, very accurately, is a very strong filter for deciding whether or not a peptide sequence matches to this spectrum. As a result, we no longer need to rely upon the precursor mass being highly accurate to limit the number of peptides overall to be compared to the spectrum. So in a large way, this isn't just a bioinformatics innovation. This bioinformatics innovation depends upon the high discrimination afforded by highly accurate measurements of mass for fragment ions. So what does OpenSearch promise us? We start with the fact that we're going to be using wide precursor tolerance that allows us to match a huge number of sequences that have very large precursor mass differences with what this particular tandem mass spectrum represents. OpenSearch claims two different things here. Number one, they claim that they're going to be able to identify a much larger fraction of your collection of tandem mass spectra than what narrow search does, or closed if you want to use their term. I don't. It's okay. So they also claim that they're going to recognize a far broader palette of post-translational modifications than you could with database search. So more of your data explained, more PTMs recognized. What's to lose, right? I would have to say that both of these claims should be taken with a pretty large rock of salt. The idea that far more spectra from data sets are identified at the same error rate is sketchy. Maybe a 10% or 20% increase, that is certainly likely, and I would believe that. If someone tells you their OpenSearch identifies 50% more spectra than narrow search does, tell them you're a filthy liar. You're allowed to do that. All right, now, can you identify far more PTMs than what you can with closed search, or with narrow search? Yes, assuredly. That's the whole point with open searching, that any mass that falls within this window you set, whether it's plus or minus 100 Daltons or plus or minus 1,000 Daltons, will be considered in comparing to that. So that is certainly feasible. What do we find might be another interesting question to get into. How do we make this fast enough? You might think to yourself, if I'm suddenly I just went through this example where we said if we compare more sequences to each spectrum, the search is slower, right? We saw that huge gain in going to semi-triptych searching or going to unconstrained searching. It became slower because we were comparing more peptides to each spectrum. Why is OpenSearch immune to that? One of the chief things that's come about is the use of fragment ion indexing, which is to say that if we know a tandem mass spectrum has a peak at 284 m over z, we can very, very swiftly determine which peptide sequences in the entire sequence database would contain an ion with intolerance of that. That's pretty powerful. Fragment ion indexing makes it possible very rapidly to discern which of all sequences that could be compared to the spectrum are truly have a match to a given spectrum. So fragment ion indexing moves us forward. Two big search engines have really stepped forward, I think, and there are others that also allow for open search. But I would highlight MS Fragger, which is a software suite that comes along with FragPipe from the Alexei Nesvishky group. Is very, very fast and is, I would say one of its special powers is that if you have lots of RAM in your computer, the software will use all of it to make the search go just as fast as humanly possible. So very impressive software. OpenPFind is another one of these fragment index-based systems. They also make use of sequence tagging to some extent, and has a strong reliance on machine learning to maximize how many PSMs it can claim at a given false discovery rate. So both of these toolkits are available for free use, at least in academic purposes. So very, very nice software. So what are the cautions that come out of this? Certainly, an enlarged search space requires greater conservatism in PSM filtering. If you are using lots and lots of tricks that sort of take us outside the standard target decoy FDR assessment, you need to be sure that none of those changes cause violations to the target decoy rules. And some of these systems, I think, are better at resisting that kind of mistake than others. Okay, next, the open search is hypothesis forming. Do not use it as your final search. This is something where I probably differ from a lot of other bioinformaticists in my field, which is to say that I feel that when we use open search, there are so many different mass shifts that come into play that the false discovery rates are not really what our software says it is. So what you should look for are patterns of modification that may be detected through a search, those patterns may inform you how to do a final database search. Use that by all means. But certainly don't fall in love with an individual tandem mass spectrum that's been matched to some modification that your PI thinks is really cool. You're looking for hundreds of hits to a particular mass shift before you could include that in a final database search. The other thing is that if you're going to distill the results from OpenSearch into something that you can apply in everyday Sequest or everyday Mascot, you're going to have to use a little bit of thought and interpretation. I'm going to give you an example of that here. Let me see. This is a different slide than I thought it was. Oh, sorry. Okay, well, we're going to get to that in a minute. All right, so we're going to start by looking at controlling false discovery rates in PTM searching. We're going to come back to this OpenSearch issue in just a minute, so hang fire on that one. But I want you to consider the problems of controlling false discovery rate when we do phosphopeptide searching. We already said serine, threonine, and tyrosine are allowed to gain 80 mass units in the case of a phospho search. We've also allowed for methionine oxidation as well, but we're going to leave that aside for the moment. So we've run a titanium dioxide peptide enrichment column, which is to say we used a column to pull aside just those peptides that contained phosphorylations, and we should have enriched very strongly for that. The great majority of the peptides we can identify from this result should be phosphorylated. That is not true of a non-enriched search. Okay? So I'm using a data set from Proteome Exchange from the LAPEC group, PXD006230. These are low-resolution tandem mass spectra with accurate mass precursors. So having run the search in MS Fragger, I ran them through ID Picker, which is a pretty conservative, fairly typical target decoy-based system. We see that we applied a 2% peptide spectrum match false discovery rate globally, which is to say that if we look at a given spectrum plucked at random out of here, it has a 1 in 50 chance of being a bogus one. We consider that I consider that an acceptable error rate overall. What we see is that 160 different peptides are observed with 3 phosphates on board. 1,237 are found with 2 phosphates on board. 7,549 are found with 1 phosphate on board, and about 3,259 are found with no phosphates on board. So we might start with that first observation, that we use a titanium dioxide column. Doesn't that mean everything should be phosphorylated? And what we see is no. Out of the set, I forget what the total of this is, say 12,000, something like that. Out of roughly 12,000, about a quarter of them had no phosphates whatsoever. That's going to happen, so we just have to deal with it. Next, I want to note that the more phosphorylations that the software was allowed to put onto the peptide sequence, the greater the error rate. This might not seem sensible. We applied a 2% overall PSM false discovery rate. Doesn't that mean that the error rate for non-phosphorylated and singly phosphorylated and doubly phosphorylated peptides should all be 2%? That's not what happens. In effect, allowing a global false discovery rate of 2% is frequently going to produce a case where the most modified peptides are the most likely to contain errors. So when I look at the peptide sequences that fall out of this, I can ask what fraction of all of the unmodified peptides could only be explained by a reversed protein entry. And what we see is that that number is below 5% for our unmodified peptides. Now the most common set of peptides were those that had 1 phosphorylation. When I look at those singly phosphorylated peptides, I see that their error rate was even lower than the unmodified peptides. So that's really good news, I think. We can trust these 1-phosphate identifications to a very high degree. But look what happens when we look at the peptides that had 2 phosphates on board, or 3 phosphates on board. The error rate among the 3-phosphate bearing peptides is almost as high as 20%. 20% of the 3-phosphate peptides could only be explained by a reversed peptide. We know those are false. So, if you see that your search engine has put a whole pile of modifications onto an individual peptide to match it to this spectrum, you had better hold in your mind that I'm not sure I can trust that. This leads to the rule warp, spindle, fold, and mutilate. Warp, spindle, fold, and mutilate is a very, very important rule to keep in mind anytime you are working with a search engine that can put multiple mass shifts onto a peptide. Allowing your algorithm to alter the peptide structure significantly by throwing on arbitrary mass shifts will produce false matches by distorting sequences with PTMs. In other words, when you start allowing the software to muck about with the sequence quite a lot, you will find that the software introduces spurious mass shifts in order to force fit an unrelated peptide sequence onto a tandem mass spectrum you've observed. That is the crux of warp, spindle, fold, and mutilate. The more degrees of freedom that you give a peptide spectrum match, the more flexibility the software is allowed in forcing a peptide sequence to match to a spectrum, the greater the proportion of false discoveries at a given score. It's very, very hard to beat that kind of principle. So when we allow the software to introduce modifications or mutations to force-fit peptide sequences, a higher false discovery, a higher rate of error is a likely outcome of it. The peptide spectrum matches that bear the most PTMs, the ones that contain larger numbers of post-translational modifications, are the class with the highest false discovery rate. You can take that to the bank. So let's talk about blind search results. Now, blind search is a term that frequently is used in the context of sequence tagging, but it's just as applicable in the context of open search. What it means is that we've gone looking for mass shifts or post-translational modifications on peptide sequences that may allow any one spot on this peptide to be modified in some way by some single mass. Okay? So there's one delta somewhere on this peptide, and it can be allowed at any position. Sequence tagging does this very well. OpenSearch does the same, but typically the result from OpenSearch is going to require some interpretation to say which amino acid bears this mass shift. Okay? So at the right, I'm showing one of these mass shift graphs that have resulted from, in this case, a formalin-fixed paraffin-embedded sample. This was one from the Nair dataset that appears in a paper of ours from this year, actually. So we're looking at the direct result of the delta mass HTML document produced by the philosopher, by FragPipe, having launched Philosopher on a bunch of MS Fragger IDs. What we see, typically, is that there are a number of peaks that show up around 0. This is to say that at 0, we have the set of spectra that can be identified to a peptide sequence with no PTMs beyond those you've pre-specified, like oxidation, say. When we note that there are other peaks around that, we note that occasionally the instrument makes a mistake in saying which peak is the precursor ion, which peak is the monoisotope that I'm going to select for this tandem mass spectrum. If it misses that by 1, we get these little off by 1 errors that result in a whole series of peaks around 0. We also have these 2 spikes at 16 and at 57. You might say to yourself, why do we care about 16? We already know methionine can gain oxygen to produce a mass shift of 16. The thing is, we told the software methionine could oxidize. It wouldn't color that in orange here, it would color it in blue because it was a pre-specified modification. These gains of 16 are all coming from hydroxyproline instead. So we didn't include that as a pre-specified modification. The software has dutifully added it as one that it frequently observes. We also see a bunch of plus 57s. And again, you may say to yourself, why do we care about plus 57? Of course, cysteine is going to gain 57 when you alkylate with iodoacetamide. But we told the software that. It found a whole bunch of extra plus 57s that are not associated with cysteine. They're associated with the N-terminus. So in this case, we see that we have a in vivo modification discovered from OpenSearch, and we have a chemically introduced modification that resulted from too much iodoacetamide. Okay, so that's the kind of interpretation that we need to be able to do when we deal with these. One of the frequent visualizations was created all the way back in 2006 by Savitsky et al. We've picked it up in some of our software, but it's just a common way to visualize mass shifts now. Each column represents a different amino acid. Each row represents a different delta mass to say this much mass was gained. So in interpreting this, you might say, well, let's start with something we know well, methionine oxidation. If we look down the methionine column, we see that the largest number is in this row. If we follow it to the left, we see that it represents 16 mass units. So we can see that the software sees a huge preponderance of tandem mass spectra in which 16 is gained there. It is not, however, the only place where 16 is seen. This is another case where we were working with a sample that contained a lot of extracellular matrix. If you follow the 16 row all the way over, yes, you see that the highest number is at methionine, but the next highest number is at proline. So this is a case where collagen present in the sample contained a large number of hydroxyprolines that led to mass shifts there. So this kind of interpretation is a very common way for people to try to make the strongest association possible between a mass difference and an amino acid that explains the most common result. You do not need to go chasing down every cell that has a number here. What you're looking for are broad patterns of modification that are characteristic of large numbers of spectra. You will take time to gain expertise in how you interpret these values. So I'm going to start with what sadly seems to be some sort of tab rule. A boring explanation is more often correct than a brilliant explanation. I'm sorry, but it's true. There are lots of times when you find something that strikes you as something that's going to go first up in a Nature publication and your career is going to be made. And yet, a chemical explanation that's much simpler, that doesn't require a new mechanism out of biology, is frequently available to explain away your great finding and your Nature paper just crumbles. Better that you should discover that than that your peer reviewer should. So, let us examine the case of a plus 22. If I see a D in my amino acid sequence, that is aspartate, I could say, this aspartate has mutated to become histidine. But it didn't. What happened was that I had a little bit of salt in my buffer, and that salt dissociated to create sodium ions, and some of the peptide ions were not bearing extra protons, they were sodiated instead. It bore a sodium on board. That caused them to gain 22 mass units. Blind post-translational modifications are useful for finding patterns of mass shifts. But do not put your faith in those individual tandem mass spectra to say, this one mass shift is one that I think is fascinating. I'm going to ignore all the other evidence and go chasing after this. Oh, you will waste so much time if you allow yourself that luxury. Unusual cleavages can appear as peptide terminal mass shifts. What on earth am I talking about here? Well, you might think that because you use trypsin on your sample, that all the peptides observable in that sample end at arginine or lysine, and the N-terminus represents another cut by lysine, by trypsin rather. What we find though is that biological samples often contain their own proteases. And, very importantly, in the process of putting 3,000 volts through a peptide, you may have broken some of its bonds along the backbone, which means that you see in the tandem mass spectrum a peptide that's missing its N-terminus or missing its C-terminus. In a case like that, these things can look like really big mass shifts or big gains or big losses at the N-termini. The Nesvishki group has created tools like Crystal C as one way to prevent yourself from running afoul of overinterpreting data like that. So there are lots of reasons why when you look into these tandem mass spectra matching to PTMs that you may have to throw away your big nature paper because there's a much simpler, much more boring explanation for why the spectrum looks that way. So site localization is the topic that I feel is so strongly associated with OpenSearch in particular. So, site localization began with people doing things like phosphorylation searches. Algorithms like A-score, I think that was the Beausoleil paper, were created around people doing phospho searches and trying to evaluate how sure they were about the positioning of the modification. Think back to that peptide we were looking at from alpha casein. If you knew that the peptide was bearing one phosphorylation, how much evidence would you have that the phosphorylation was on a serine or on the threonine right next door to it? There's not a lot that differentiates those two scenarios. If the serine is phosphorylated or the threonine is phosphorylated next to it, most of the ions remain in exactly the same position. It's only the pair of ions that differentiate those two positions that are going to shift in response to it. So anytime we interpret these tandem mass spectra with some assertion about the post-translational modifications on board, we're going to have to ask ourselves the questions: how sure are we that this mass shift is located here and no other place in the tandem mass spectrum? Those have come about via a wide variety of ways, but generally speaking, we find that some folks have introduced new scores, such as the A score, for asking how much better does this match when the mass is here than here or here. That's a rescoring, essentially, of a PSM to ask about the confidence of its location. We also see that people use delta scoring, which is to say that if the search engine reports multiple positions for that modification and provides a search engine score for each of them, you can ask how much better the score is for the top rated position than for the second best or for the third best. So you don't have to rescore it, you're just using more scores that were also output by the search engine as a way to evaluate your confidence that this location and this location only is the way is a sure bet for interpreting the spectrum. So there are lots of techniques now for identifying and verifying post-translational modification data. Stepping beyond database search is good because anytime you're trying to look for a whole palette of modifications, you really need tools that are going to support that better. Resisting false discoveries is essential because post-translational modification search spaces are much bigger than standard ID search spaces. It's not just going to take more time to run the search. It's going to require it's going to give you a sensitivity hit in losing some identifications that were on the edge with standard protein ID that fall away as soon as post-translational modifications broaden the search space. Biological databases can contribute lots of perspective to evaluate the reasonableness of a post-translational modification. If you see something that looks like a plus 12 or a plus 46 or whatever, you can enter that mass shift into delta mass or just browse to that location in the mass table to give you some idea of different possible explanations that exist for why this mass shift would be seen in a particular PSM. So I hope that these rules have helped you to know how algorithms can help you and also what kind of skill set you're going to need as you interpret the results from post-translational modification. Happy hunting!